everyone, and welcome to another very exciting seminar in our online Evolution and Ecology series. My name is Julia Darolzi, and I'm a postdoc at the University of British Columbia. Myself and the other organizers, Elizabeth, Andreas, and Wouter, are delighted to have with us today Professor Jessica Metcalf from Princeton University. Professor Metcalf is an expert in disease modeling and population research, studying the impact of demographic change on human health and disease incidence and spread. Professor Metcalf's work is also focused on understanding the fundamental drivers of immune function, as well as immune trade-offs and their consequences for sex differences in immunity. Before I give the floor to our speaker, I just want to quickly remind everyone watching that, as always, there will be a Q&A session after the talk. If you would like to post questions for the speaker, you can do so in our designated Slack channel, where you can also upvote the questions that you would like to see answered. So with that, thank you very much, Professor Metcalf, for accepting our invitation, and over to you.
Well, thanks very much, and thanks for the organizers for organizing these. I've really been enjoying them over the last couple of weeks. And happy Malagasy Independence Day, which is what it is today. So today I'm going to talk about sex differences in immune function. And I'm going to focus on sex and not gender, although gender can also affect sex outcomes. And the first thing you learn when you start reading about sex differences in immune outcomes is that we know astonishingly little about sex differences in immunity. In a lovely review, Sabra Klein, who's one of the absolute pioneers in this field, pointed out that in 1977, the Food and Drug Administration explicitly prohibited clinical trials on a woman of childbearing potential. In 1993, they decided that women might be human. But even in 2005, 8 out of 10 drugs from the market results in women. So there's just an astonishing gap around what immune function looks like, even in our own species. And of course, if you start digging into that research history, not only is this curious gap present, but you also discover the kind of social and cultural maelstrom of people banding around words like the weaker sex. It's very interesting. And so what I'm going to do today is I'm going to talk through an example of how sex differences might shape outcomes, taking a focus on influenza in our own species. And then I'm going to dig down and think about how sex differences in immunity might evolve. So it's stepping back and thinking about the bigger picture. So influenza is a remarkably intricate challenge for our immune system that plays out in complex ways across our life course. And this is mostly because the virus evolves. And so the pieces of the virus that our immune system sees, the neuraminidase, the hemagglutinin, tend to change from year to year. So our immune system has to kind of keep up with that challenge. Work from my colleague Justin Lessler looked to the immune system response of a large cohort in Guangzhou province. So what this graph shows you on the x-axis is different circulating flu pandemic strains, and on the y-axis, the age of individuals. And the important piece is that the darkness of the colors is a measure of antibody titers, the pieces of the immune system in your blood that can recognize that particular virus. And so just to illustrate how this works, if you look on the far left of this plot at Hong Kong 68, this was a viral strain that circulated around the globe in 1968. Then individuals who weren't born at the time this virus was circulating, so individuals who are aged between 0 to in their 20s at the time of the survey, which was sometime in, I think it was 2016, show no antibody titers to this. And why would they? Their immune system hasn't seen it. They have not developed memory to it. There's very little response. As you move further forward into the future, as you move through Victoria 75, Bangkok 79, you can see that younger and younger individuals are beginning to show responses to this virus. There's darker areas on this plot. So it's really fascinating, but you can also sort of potentially see that it's not just that if you're too young to have seen it, you don't respond. There are also differences in the magnitude of the response. And Justin and colleagues pulled together a very simple model to try and probe this. And what they showed was that it's not just how old you are that determines the magnitude of your response to a circulating influenza strain, but it also depends on what age you were when you first saw that influenza strain. So this is what's shown on this plot here. They basically fitted a model that had an intercept for different viruses, an effective age, your age, and the circulation of that virus. but also, and your age in absolute terms. So what I've shown you here is the piece on the age at viral isolation. And it's kind of fascinating because what it tells us is that our immune system is completely obsessed with whatever virus you saw when you're around age 7. So whatever virus popped into the world when you're around age 7, that's the one your immune system thinks about for the rest of your life. And it's probably got to do with this process which has been described as original antigenic sin or original antigenic seniority, where something about that first hit of memory is the thing that sticks. And of course, I think this is very interesting, and it's very interesting thinking about how the immune system works in very general terms, but the title of the talk perhaps gave you a hint as to where I'm going to go next. And the obvious thing to do with this model, which crisply described processes, at least the phenomenological aspect of processes that probably are important in shaping our immune system over life, was to include sex differences. So all I've done here is exactly the same model, but adding into it sex differences as a covariate to try and explain the magnitude of the antibody titers observed in individuals through time. Okay, so how much your immune system is reacting to different influenza strains. And what this lower plot shows is, again, on the x-axis is your age at viral isolation. The upper plot shows the mean as before, but the lower plot shows the difference between the sexes. And so the lower half below the horizontal line indicates that the female effect was larger, and above the line indicates that the male effect was larger. And so what you can pull out is that females respond more to viruses that were circulating before they were born. So the x-axis values of less than 0 indicate that that virus was circulating before the individual was born, which suggests that female immune systems are more cross-reactive. So they tend to see things, they don't react just to the specific shapes that they've seen, but they kind of react to shapes around that. The other piece you note is that as you get to around age 45, again, we see a much larger effect in females. And if you're an evolutionary biologist, one of the things that you anticipate is that everything should kind of fall apart at late ages. It's senescence, the evolution of senescence is one of the kind of strong predictions of basic life history theory. And so this might suggest that males are falling apart faster than females, at least in terms of their antibody responses to influenza. But it's not clear that that's the case. Although it lines up with our expectations, so we know that females are better, at least in terms of life expectancy and survival. We tend to live longer than males, but we don't know whether antibody titers per se translate into higher survival. And that's the key question, right? This bigger antibody titer might mean you're better at controlling flu, but it might not as well. An immune system is a dangerous thing. We don't actually know exactly what antibody titers translate into in terms of health outputs. Fortuitously, as part of this work, which is part of the Fluscape study that has been running for a number of years, they had collected biobank data and a nested case control study of 100 sex and age-matched deceased individuals and living controls. And they looked at the antibody titers of these individuals across the full spectrum of circulating influenza strains. So just as shown before, but now with the added metric that we can tell what these particular titers did to survival. And what we did was we just fit a logistic regression to test for an association between survival status and influenza antibody titer using conditional logistic regression. And so I'm going to show you the results here. On the x-axis is the year of circulation of the virus strain. And on the y-axis is the log odds of an increase in mortality. So above the horizontal line there is bad. On the horizontal line means there's no effect. And it starts in the first year of the appearance of H3N2, which is one of the influenza viral strains that has been going around the world for a while. And this is what the results look like if you just fit them to all the data generally. And it might not look very exciting to you because all the standard errors overlap with 0, but the study was not powered for conventional significance cutoffs. It was a pilot study. And it is still kind of amazing, given that this was a pilot study, that the magnitude of effects suggested, at least. A 20% reduction in mortality for 1 log increase in antibody titers is what is suggested by that far left point on this plot. And it seems very unlikely that this is about influenza itself, because influenza alone could not cause quite that much mortality. It might be that it's a more generic measure of immune system function that we're capturing somehow. So it might be that, yeah, influenza antibody titers are correlates of more general protection. But of course, again, you know what I'm going to do next because of the title of the talk, which is to take the same model and break it apart by sex. And here you get this very striking effect where a single log increase in antibody titers in males is almost always protective, reduces the mortality, whereas in females, if anything, they're above the line. Again, this isn't powered, but it's remarkably consistent, and you can kind of bootstrap across all the different strains and show that this pattern holds. So how could that possibly be happening? If we assume that there's some relationship between the antibody titers on the x-axis and survival probability every year, then it could simply be that this is an issue around nonlinear averaging. So females might just have higher antibody titers in general. This has been observed to be the case. And they might also be in this higher survival space. And if then you try to map back to survival probability, what you'd find is that males had a really big difference in terms of the antibody titers relationship with survival, whereas females had a very small one. So it could just be the fact that females are on this higher part of the curve that is obscuring the relationship, if the same relationship holds for males and females. Of course, it could be that the same relationship does not hold. There could be some other driver acting in females. They could be bringing more weapons to the war in some sense, in which case, again, you'd have the same sort of pattern. Or finally, perhaps the most extreme potential is that there could be literally a different relationship between antibody titers and survival in males and females. It is clearly relatively straightforward, actually, to obtain data to narrow down on which of these different effectors it is. The data is already, much of it is collected, it just requires testing. Unfortunately, it's sitting in Guangzhou province in China, where our co-authors and collaborators are rather tied up with other things at the moment. So I don't expect this work to move fast very soon, but you can clearly see that there are clear applied issues, especially given that one of the things we do with our vaccination programs is push around antibody titers. And so this has the potential to push around impacts of health differentially in the different sexes, which would be of interest to try and pin down. So that's a very broad motivation. There's also another clear broad motivation in the current landscape of global health, which is COVID-19, and I can come back to that later. But it's clear that these sex differences in immune function exist, and it is also the case that they probably have implications for health. So the next question one might ask is, why? Why would sex differences in immunity evolve? What might have driven, what might be the ultimate processes underlying these sorts of patterns? And if you're an evolutionary biologist, the explanation is relatively straightforward, or at least the conventional narrative is straightforward. Males are the weaker sex. They invest in fabulous displays like this peacock tail, antlers on deers, weapons for competition. And the argument is that this results in shifting resource allocation away from immune function, say. The conceptual bones were put on this narrative really very early on. So Angus John Bateman published an experiment in 1948 where he mated flies across multiple generations and kept track of male and female fitness. And he concluded from this work that male fitness increased with the number of matings they secured, whereas female fitness increased with the number of times they reproduced, which tied in with their longevity. The thought was then that males should invest away from immunity and towards securing matings. So far, so simple, but this is clearly too simple. And there are many issues in this work which were, I think, teased apart very beautifully by Hannah Coco and Jenians, where they pointed out that, for example, more immunity doesn't necessarily translate into greater longevity. There's many other potential sources of mortality. And of course, longevity doesn't necessarily translate into higher fitness. Fitness is going to be a compound of both survival and reproduction. And the moment you start reading into how the immune system actually works, so what the kind of knowledge of the mechanism tells us about these processes, you learn that it isn't just, you really can't simply stick with the concept that males are weaker. There's lots of other things going on. And so what I wanted to do next was to turn to what we know about our own species. This is Justin Lesser's baby. And to think about what we know about the species where we know the immune system the best, and see if we can translate that to thinking about the evolution of immune systems more broadly. And of course, this is the other way around from which we normally go. Normally, we start with model animals and try and learn something about human health. But in this particular case, the breadth of information about the immune system in humans seemed to at least provide a starting point for thinking about the broader evolutionary context. Of course, you have to think quite hard about what humans are in the context of the larger tree of life. And we're sort of normal in many ways. So we're mammals, which means we have determinate growth. We're senescent. We have to care for our young. We're primates, so we're big-brained and long-lived mammals. That's also pretty consistent. But we're really odd primates, as was pointed out by my colleague Samuel Pavard. We live twice as long as you might expect based on size. Our maternal mortality is incredibly high, and our birth intervals are very short despite a long altricial period. And then, of course, there's menopause, which is another really funky piece of human evolution, which is really, you see in maybe a few cetaceans, but that's about it. So keeping that in mind and moving forwards, what are the two root causes of differences between the sexes? The first is chromosomes, obviously, and the second is hormones. This is the karyotype of a human male. And if you narrow in, of course, a human female would have two X chromosomes instead of the XY. And again, as you start reading about this, it's absolutely remarkable how many genes involved in immune function sit on the X chromosome. So there's many sort of classic pattern recognition receptors which are involved in detection of pathogens that only sit on the X chromosome. And since, as Mary Lyon worked out, is one of the ways in which males and females end up with the same gene expression, at least in humans, is that one of the two X chromosomes in every cell is silenced, but it's silenced at random, which means that females are much more diverse than males in terms of these particular genes. And it also is the case that often pieces of the X chromosome avoid silencing. So in particular, things like toll-like receptor 7, this is one of the classic pattern recognition receptors, seems to have higher expression in females, partly as a result of its escaping silencing. So it's got twice as much per cell. And that's been shown to be protective in terms of influenza, but perhaps detrimental in terms of autoimmune diseases. It's worth keeping in mind that this sort of foreshadow is one of the really important things I think you have to think about when you think about the evolution of immune function. The immune system is both good and bad. So it's both protective against infectious diseases, but it's a very dangerous line of defense because it involves often essentially destroying your own cells. And so that's obviously a dangerous thing to be playing with. It's also worth keeping in mind that this XY setup that we mammals have settled on is not the norm necessarily. So this is some lovely work by Pipoli and colleagues that looked at survival, the adult sex ratio, which is a measure of survival to adulthood across species with different sex chromosome setups. So the classic difference from us is birds. In birds, males are not the heterogametic sex, females are the heterogametic sex. And in tetrapods, it's all over the show. So sometimes it's males, sometimes it's females. It's like it's a very pleasing mix. And what they showed in this paper was that, yes, being the heterogametic sex does seem to always be associated with a deficit when it comes to the adult sex ratio, suggesting that you do pay a survival cost, regardless of whether you're male or female, just from having that one tiddly chromosome, which can result in recessives being revealed, etc. So that's the first piece. Chromosomes will define one chunk of sex differences in immunity. The other piece is, of course, hormones. And this is something that I found really surprising, but probably isn't that surprising for anyone who's thought about this for 2 minutes. Every single one of our immune cells is covered in hormone receptors. And that means that estrogen receptors, progesterone receptors, androgen receptors are shaping what happens in the promoter regions of genes in innate immune cells and adaptive immune cells. Every part of the immune system is constantly being tuned by our hormones. So you've got to think about sex differences and you've got to think about hormonal differences as you try to think about what might shape the evolution of immune systems and sex differences in immune systems specifically. If you're an evolutionary biologist, what you need is not just the mechanism, you need to think about the trade-offs that might be governing where investment should lie. So there's many ways to think about this, but the way I ended up breaking it down was in terms of thinking about investment in detection or discrimination of pathogens. And then once you've detected or discriminated the pathogens, which is arguably quite a hard piece, then you have to respond to them. And in order to appropriately counter the thing that you've detected. And if you start reading through the immune system literature, first of all, you become extremely depressed because it's unbelievably complicated. But the various lines of evidence at least suggested to me that you could build a cartoon of how the immune system worked, where females seem to be investing a lot in the things that line up with detection and discrimination, whereas males are lining up with the pieces that line up with response. So this won't mean anything to you unless you're an immune system nerd, but the CD4 to CD8 ratio is higher in females. Immunoglobulins are higher in females. These are the sort of pieces that respond to 30 bits of virus. T-cell activation, some of these pattern recognition receptors, the bits of the immune system that present bits of virus to the other bits of the immune system are much more active in females. All these pieces suggest that they really, the surveillance piece is really working extremely hard in the female immune system. Whereas in the male immune system, what you see is more natural killer cells. That's what NK stands for, which they do what their name describes. They just go around killing other cells. CD8 cells, which are cytotoxic cells, which is another way of saying they go around killing other cells. And then macrophages and neutrophils have more TLR4 expression, which suggests that they might respond more heavily to any trigger. So my cartoon of the immune system, and you can, it's surely an inadequate cartoon in many ways, but suggests that if you looked in detail at all of these drivers, you could at least broadly group these as suggesting that females are investing in detection and discrimination, whereas males are investing in response. So what does this mean in terms of trade-offs? So if you think about a model of how these things might work, the challenge that the immune system faces is quite straightforward. There's some set of molecular patterns or signatures that are indicative of self, and there's some molecular patterns or signatures that are indicative of non-self. And I've put as an illustration there on the x-axis CpG content. So for reasons to do with methylation patterns, etc., we tend to have very low CpG ratios, so C's followed by G's in our genomes. And there is a pattern recognition receptor that just goes around looking for high levels of CpG ratios and identifies that as being bad. So that's a very simple way, right? Like you would know that anything above a particular threshold was non-self, thus likely a pathogen, and thus should probably be attacked and evicted. In this particular instance, if you had to draw a line separating good from evil, you would draw it down where that dotted line was, and there would be no problem. But of course, the world is not like that. And one of the things that happens is that pathogens will evolve to mimic us. And this is something that's been documented, for example, or at least suggested to occur at least in influenza, where they evolve to have CpG ratios that look more like us. So whatever this continuous variable is that distinguishes self from non-self, we actually expect pathogens to be evolving along it to look more like us so as they can evade detection. And that immediately raises a problem, because now you have the situation where wherever you draw the line, you're going to have a certain number of false positives and a certain number of false negatives. So everything to the left of the line is going to be things that you don't respond to. And everything to the right of the line is going to be things that you do respond to. And so responding to things that aren't actually pathogens bears the cost of immunopathology, whereas not responding to things that are pathogens bears the cost of disease-related mortality. If you squinch that vertical line to the right, what you do is you increase the number of false negatives, right? There's going to be more of the red bar to the left of that vertical line. Whereas if you squinch that vertical line to the left, you're going to have more false positives. You're going to be responding more often to your own self, attacking your own self, destroying your own cells, even in the absence of a pathogen. This is something that epidemiologists think about a lot, and the way because of everything from trying to define who's diabetic to who isn't, many discrimination questions around states of disease rely on these shaky measurements. And the way they frame it is in terms of sensitivity versus specificity. Sensitivity is the probability of identifying a true positive, whereas specificity is the probability of identifying a true negative. And the curve, there's a sort of trade-off curve that looks like this. So if you were to place your arrow all the way over to the right, what that means is that you're very rarely getting it wrong in terms of triggering in response to yourself, but you're missing a lot of the pathogen. So your sensitivity is low. And as you move the arrow over to the left, you increase your sensitivity, but you reduce your specificity, and so on. If you're all the way over on the left-hand side, you're going to have all your negatives are going to be true, but a lot of your positives are going to be false. So it's a pretty, if you're an evolutionary biologist, this is immediately titillating because it's a very crisp trade-off, which means if there's a crisp trade-off, you can start looking for what the sensible thing to do might be. And my expectation based on plowing through the immunology literature is that I think that males should be more on the specificity end. So they might be missing a lot of the pathogens. They don't have huge amounts, they do have huge amounts of selection on self-reactive T cell and B cell receptors. They don't have as many antibody presenting cells. I expect them to be less sensitive. Whereas I would expect females to be more sensitive in terms of the magnitude of their response. And this is because they have more active toll-like receptors, these pattern recognition receptors, they have more efficient antibody presenting cells, all these sorts of pieces. So that's the first trade-off. It's just finding the how much you respond in terms of triggering an immune response, how much work you do to detect and discriminate between self and non-self. The other piece might be in thinking about the magnitude of the response. So once you've decided to go for it, that this is non-self, that this is the enemy, then you can think of a spectrum of response, large response on the left-hand side of this plot, suggesting you deploy huge numbers of immune effectors, macrophages, other important immune cells, which will definitely reduce your risk of dying from infection, but will also increase your immunopathology. So your own cells that are killed by your immune system are likely to increase in those settings. Whereas conversely, if you didn't throw too many immune effectors at the problem, you wouldn't suffer from immunopathology, but you might also not be terribly good at controlling the infection. So you can see a trade-off there, either paying the cost of immunopathology or paying the costs of infection-related mortality. And again, I think that based on the fact that males have all these cell-killing immune cells in the mix, or at least higher numbers than in females, I would expect males to be up at the top end of that scale. Okay, so what I'm going to do now is put these two things together, and my expectation is that because they are clear trade-offs, there's going to be some intermediate optimum. There's going to be a level of, as you move up in terms of sensitivity, you start by increasing the degree to which you protect yourself from pathogens, but then you go beyond a certain point and that is outweighed by the fact you're attacking yourself. So there's a hump somewhere in the middle. And the same in terms of how many immune effectors you throw at the problem. And these two things should depend on each other. And you can write down a very simple model in terms of hazards that describes this. And this is what I've shown here. So on the x-axis is going to be that magnitude of response, how many immune effectors you throw at the problem. And on the left-hand, on the y-axis is going to be the degree to which you're working on discrimination. So whether you are going in the direction of false positives or false negatives. So you can lay it out, and this is what it looks like. The white areas are the areas of highest fitness. And what you find is that, if you're extremely sensitive, so if you respond all the time, then it makes sense not to have really massive responses. This kind of makes sense if you think about it. It's pretty intuitive. Whereas if you're not that sensitive, you might as well go big whenever you do eventually respond, because it's very likely to be a pathogen. So that's nice. It tells us that it's very straightforward to get two optima in this relatively simple setup with true trade-offs. Not that surprising, but it doesn't actually answer our question. You can't just look at this and say, well, two optima, two sexes, let's just stop there, because it's not clear why females should be on one optima and why males should be on the other. So we went back to think about this again in the context of an animal. And again, one of the basic assumptions was that within this larger context of these immune trade-offs, males might also not be spending so many resources on immunity. That's a crude first pass. Of course, there's a lot of complexities in there as well. But if we were thinking that males are just cheaper in terms of how much they spend on immunity, how would this play out in pushing around this fitness landscape? So if we frame it in the context of these two trade-offs, if males are not investing so much in the immune system, it might be that their discrimination is sloppier. So they just don't have as many cells or as refined cells looking for that discrimination between self and non-self. And so you could conceptualize this as those two distributions being more overlapping from the perspective of males, which is very easy to implement within a model. If you thought that it wasn't so much the discrimination piece that males were allocating resources away from, but the magnitude of response piece, then you would just simply say males are maybe throwing less immune effectors at this problem. The trouble is you can put this in and there's like a nice analytical solution to it, and it just doesn't work. So none of this results in males being the less sensitive sex. It really troubles me that that's how this framing works out. But nothing in how you frame, if you reduce allocation to the immune system in one of the two sexes, then that does not result in that sex being less sensitive. So immune trade-offs are not going to be the answer there. Is there another thing we can think about? Of course, there's another thing we can think about. Life plays out across age, and males and females are making strikingly different investments at different ages, and they're undergoing different risks. My favorite definition of life history evolution is that it is an elaborate answer to the simple question of why having more offspring is not always optimal. And I think it's a de Jong paper. And once you start thinking about that, so putting these immune trade-offs not within allocating away from immunity generically, but start thinking about what it means across the life course, a little more information pops up. So to do that, one of the best ways to grapple with life history is to build a demographic model. So very simple idea. You start off with survivorship to every age. You multiply it by fertility at every age. Offspring at any age, you have to survive up to that age. And you sum that product and you end up with fitness. You know, the concept of R0 is probably giving people whiplash at the moment, but it's much more relaxing if you think about it in terms of elephants. It's not doubling every week. And so with this, you can figure out what the optimal strategy might be, thinking about how these investments might play out at different parts of the life cycle. So in the specific case of males versus female mammals, we can think about different effects of pregnancy. So higher mortality might be one outcome. Higher infection might be something. Your immunity might be suppressed. You might have higher infection-associated mortality. Carrying offspring to term is a dangerous and difficult thing to do. But it's particularly dangerous in humans. I think maybe that's why I was so obsessed with this piece when I started writing it. But it's certainly one of the pieces that could drive selection in the direction of increased sensitivity in females. However, there's a counterweight, which is that one of the other ways in which immunity might play out is by resulting in greater miscarriage. I mean, one of the things that female mammals do is essentially carry a pathogen to term. So that discrimination between self and non-self, suddenly that ability to be very, very picky about rejecting non-self is not such a good thing. And my colleague here at Princeton, Andrea Graham, has done some lovely work showing that this, at least there is some empirical support for this idea in the Soe sheep, where females that survived better, that seem to have higher antibody responses in an antibody that basically responds to self. So it's like a marker of infections, of autoimmune diseases like lupus in humans. They survived better, but they had more miscarriages. So that trade-off seems to be playing out. And this is actually one of the, I think, the puzzles that first got me thinking about these differences is it just seems really odd that females should have these super picky immune systems, that 80% of autoimmune diseases, for example, are concentrated in females, when this is also, there is a certain need to tolerate non-self over the course of pregnancy. So it's quite surprising. So you can balance things out that way, and you can find situations where you have higher selection for sensitivity, but there's also pieces that suggest that there should be lower selection for sensitivity in females, like this miscarriage story. And you can also start thinking about things like if late life reproduction is really important, and this might be the case, for example, with deer that retain harems apart starting at a certain age, then selection might favor lower sensitivity. And this is because, so sensitivity means that you trigger very easily, and you trigger to self as well as non-self. You're undergoing that risk every year of your life. So if infection is concentrated early in life, this just means you're paying this needless cost over the many, many years that are important to your fitness if late life reproduction is critical. So another way in which males might be evolving towards lower sensitivity, but none of this was totally satisfying, right? It seemed like all of these arguments could go one way or another, and none of them was a kind of slam dunk. And there is an obvious piece of the story that is missing for anyone who thinks about immune systems is probably shaking their heads in despair. And that is that I've been talking about this as if it's really an innate immunity story, as if it's like you see a pathogen and you respond. But one of the most absolutely amazing things about the immune system is that we have this feature called adaptive immunity. And this is where if I have had measles, my immune system will remember that I had measles. And I will therefore be protected from the rest of my life because memory B cells are sitting around waiting for measles to come back and can generate such an efficient, rapid response that measles never has a chance. And you can see this is an incredibly powerful thing in a world where pathogens are recurrent and where you're not paying either the cost of immunopathology or the cost of infection because you've shut down the problem really quickly. So that's adaptive immunity is clearly central to any story about immune systems in mammals, but it's also, I think, particularly relevant to sex differences because because of the importance of maternal immunity. So again, if I've had measles, if I were to have children, I transfer antibodies, these things which glom onto viral particles as they're floating around and protect me from them and elicit an immune response that protects the child over the first year or so of their life. So these wane eventually and as children develop their own immune systems, but they provide this first layer of protection before the children's immune system has developed that is sort of passively transferred from breast milk and through the placenta. And so it could be, because this is something in which males have a very important piece of thinking about how the evolution of immune systems might occur. So you can build a very simple model to tackle this, where you throw a stick. I'm asking the same basic question, but because I'm trying to think about memory now, and because it's possible that I'm going to end up with more states than I have individuals, because you have to have memory to different pathogens through time, I ended up doing it with an individual-based model. And what I did was I said every individual is defined by their threshold. I threw a distribution of pathogens at those individuals. They responded according to the rules shown on that right-hand plot. And then they reproduced, and offspring inherited their mother's threshold and potentially protection. So based on what their mother had seen, they would be protected against the pathogens that their mother had seen for that first year of life. And then they're exposed to a distribution of pathogens. They die according to the hazards described, and then they reproduce again. Okay, and so if you put those things together just to make sure that it works as a kind of sanity check, you can build the fitness landscape that emerges from this just in the null case where we're not thinking about differences in offspring immune systems. And I can plot against the x-axis here, which is essentially a metric for where I'm drawing that line. Am I drawing it on the far left of the graph, in which case you're responding to everything, which means that you just have a really high burden of immunopathology, or I'm drawing the line on the far right of the graph, which implies that you're not responding to anything because your line is on the far right, and so you therefore have a high burden of disease. And I'm going to look across that whole spectrum of where you might draw the line and measure what the model throws up in terms of the probability of surviving. And of course, this depends on two critical parameters, one of which is how much you die from an infection, and the other of which is how much you die from immunopathology. And on the right-hand side of the graph is where the deaths from infection will be most apparent, because you're drawing the line so far to the right that you're not actually triggering a response to anything. And as the infection mortality increases, you end up with a lower survival probability. On the far left of the graph, which is where you're responding to absolutely everything, is where we see the biggest effects of the costs of immunopathology, shown by the straight to sort of increasingly dashed lines. And again, if you have a massive response in terms of immunopathology, if you have a massive cost of immunopathology and you're all the way on the left, you're going to be paying the highest cost. And the basic outcome is that there is an intermediate optimal across this spectrum that's defined by how those trade-offs line up. Okay, so we have that, and now we're going to take that basic premise and we're going to add a second layer to the life cycle. and that's children. And we can first assume that children they haven't got a developed immune system, and say their mothers don't transfer any antibodies to them, what happens? They have no protection. So basically their survival is entirely defined by how much the infection kills them. So as the hazard associated with infection goes up, that children die more. If, conversely, you assume that children are inheriting some protection from their mothers, what happens is that for all of the thresholds on the left-hand side, you end up with higher survival as the children are protected from the pathogens that their mothers have seen. So as you move further, as mothers have thresholds further over to the right, then they end up, that which means that their mothers are not responding to things, then the children are less protected and their survival starts going down. And the optimal for the life cycle will be, of course, some intermediate between these two. And so you would expect this to shift the optimal for females to the left relative to the optimal for males. So I think that's perhaps a more compelling explanation for why sensitivity might be higher in females than males. So I think just to sort of conclude, male and female immunity is really strikingly different, and this is likely to emerge because immunity is both protective and dangerous. There's an awful lot of space to work with around how the immune optimal will shake out. If females do map to more sensitive responses, which is of course debatable, you could argue with my interpretation of how the immune system works, then it's likely that maternal immunity might be one of the important drivers, at least in mammals. And of course, there's lots of really exciting directions for testing this. Males, seahorses are the ones that get pregnant, and my colleague Olivia Roth has done some lovely work trying to tease apart what the male seahorse immune system looks like. And it does look very different from the female immune system, more in line with what we see for, as it were, mammals. And there's, there is also evidence what other gene is what will shape what you're protected against. The transfer of maternal antibodies in pigs and mice, and even in humans, has been shown to be shaped by whether you have a homologous or a heterologous vaccine. So whether there's a matchup between the pathogen you see in the vaccine you saw or not. Just to wrap up, so what I did was I took the exemplar of influenza and thought a little bit about how sort of counterintuitive and strong these different effects could be, despite the lack of information on them, and then thought through how we might need to go beyond just thinking about a male and female immune systems as being weak or strong. Males as the weakest sex doesn't quite work. These results are summarized in a couple of papers here, I think thank you very much for your questions.
Thank you very much. That was very, very interesting. We have a few questions starting to come in. One of them is, since immune response is affected by both hormone and chromosomes, do you know whether cross-sex hormone treatments affect the immunity and how?
So that's a really interesting question. I think that probably the answer is in the work of Sabra Klein, who is just phenomenal at integrating the really interesting and diverse literature around these things. I'm afraid I don't have a crisp answer. There is some stuff on the effects of hormones that you can see through aging, for example. So testosterone declines, and of course, there's these tremendous perturbations around menopause in human females. And you do start seeing signatures of that in immune responses. But I think there's an awful lot still to be done in teasing those things apart.
Yeah, and in relation to menopause, how does that affect late age immune function? Can you say anything more about that?
So I think that's very interesting as well. So the theory of senescence basically suggests to us that everything should fall apart as you get older. The interesting thing about the immune system is that if you look at it, it looks like it's just acting up more when you get older. And I think that's to do with the fact that the immune system is this incredible self-reactive thing, right? Half the immune system is designed to shut down the other half because it's so incredibly dangerous having an immune response. And this means that once the regulation starts falling apart, you end up, and I think Alexei Metcalf has written really beautifully about this, you end up in a world where you have these sort of firestorms. In fact, people have used the word inflammaging to describe aging and senescence of the immune system. The ways in which this shakes out, I think there's an awful lot of really interesting things to be done in the context of male and female differences at late ages in many species, including our own. There's this wonderful, the demographers talk about the male-female health mortality paradox. And this is where at late ages, by just about every metric, males are in better health than females at a comparable age. So they stand up quicker, their handshake strength is stronger, they just seem to be in better shape, and they self-report as feeling excellent. Whereas females are like, I feel dreadful, and then females live forever, whereas males are like, I feel great, and then they just die. So, and we don't really understand, why that is or how that plays out. But I think that there's an awful lot of really interesting pieces around the fact that it just seems like the inflammation-y pieces are to some extent controlled by the adaptive immunity, and the adaptive immunity might be waning more rapidly in its effectiveness than all of the innate immunity, and that might lead to these sorts of states. But a crisp summary of exactly what's going on is something I'd long to read.
It's very interesting, thank you. There's another question. What pattern of immunity in males and females do you expect in species with external reproduction?
So there is transfer of antibodies through egg yolk, for example. So I think that there is some lovely work from a whole group of people whose names are now escaping me on birds and bird immunity. So I wouldn't be too shocked if there were similar effects there. I'd love to do more on tetrapods with maternal care and all these sorts of things. All the models I built have this dodge in them where I don't formally really engage with frequency dependence or mating structure. I build males and females separately, and I don't account for the fact that they have to reproduce together. So I think there's a lot of really interesting things around systems of parental care, as well as external versus internal reproduction.
Thank you. You mentioned male seahorses getting pregnant. Would this mean that their offspring inherit the paternal immunity? And is this only acquired through pregnancy, or is there a reason we don't consider paternal immunity inheritance in female pregnancy, in species that have female pregnancy?
So I would refer you to the wonderful work of Olivia Roth, but she has shown, I think she did so I may get this wrong, but I'm going to try and recreate it. She exposed mothers and fathers to Vibrio cholerae. Apparently that's just what you do when you're working with seahorses, and then looked to see whether there was immunity in the kids. So she did one parent or the other parent or both. And it turns out you inherit from all of them. Right, and mechanistically that makes sense, right? If you're you're chunking across an egg and there's egg yolk and all this stuff, so you have an opportunity to put antibodies in if you're a female seahorse, and if you're a male seahorse, they're sitting there. They have this. So seahorses, I mean, it's a whole set of the cygnathids. There's a whole set of life histories within the spectrum, but they have this very elaborate brood pouch which has gas exchange and all these sorts of things. So there's also opportunity to transfer antibodies there. I think it might just be logistically much harder because sperm, there's not a lot of space in sperm.
Someone is interested to know whether female infants or newborns also show similar patterns as adult females in their immunity.
So I think as you'd expect, so there are differences in males and female humans very early in life, but it really kicks off around puberty, potentially because of the hormonal effects. So there are these, so for example, flu is kind of funky because females die more of flu during adult years, but die less at very early ages or very late ages. And I think it'll, again, there'll be a balance of all these different factors, but from the get-go, you're going to have more toll-like. So the ontology of the immune system is its own special headache. But from the get-go, just based on what we know about chromosome silencing, etc., you'll have more as a female human small child. You'll have more toll-like receptor 7 floating around, for example. So you'll have sort of double the ability to respond to certain viruses.
There's another question. Can you extend some of the results that you've shown to response or vulnerability to cancer? What's that?
I think that's a great question. I think there's some really interesting digging to be done around the time course of cancer. So cancer is, it's a piece of self that has gone rogue. So it's hard to see if you're the immune system. and the females tend to have later, if you sort of throw away the reproductive cancers, they tend to have later onset cancers. Of course, you have to account for the fact that men tend to smoke more and where our data comes from, and that there's always behavior as well as immune system function. But yes, I think that's a rich area for thinking about discrimination in particular. So it'll be a really good lever to pull to try to think about whether my cartoons of how this works actually play out in meaningful ways.
Thank you. What's the role or importance of sexual conflict via expression or imprinting in interpreting sex differences in immunity?
What a great question. I'd love to know. I wish someone would do that.
I also had another question in trying to understand the relative contribution of sex chromosomes versus hormones to sex differences in immunity. Have there been any studies, if you know, of species that don't show sex differences in their sex chromosomes that much, and whether they're still showing immune differences with sex, that more or less than you would expect?
So there's lovely work on insects of various kinds, right, which don't have testosterone and sometimes have different genetic structures. So I think there are many paths to developing sex differences in immunity, and there's some really intriguing patterns that came out of the. So now I'm actually talking about the lack of testosterone, not the lack of sex chromosomes. I don't actually know of anything specifically on species. Oh, you know what? They're the cygnathids, which are so funky. So these are the pipefish, including seahorses. I don't think we know where the sex-determining region is yet, which implies that there isn't a clear sex chromosome story, at least. So there's one. They're particularly crazy.
Yeah, that'll be interesting to look at. I think we have one more question. There's known to be differences during pregnancy in response to male versus female fetuses. Could you speak to why there might be these immune responses so early on, and why mothers might respond differently depending on the sex of their child?
So that's a fascinating question. I think that that's a fascinating question. I think that you can walk down a Trivers Road and think about allocation in male versus female offspring and that sort of thing. I haven't really thought very deeply about that yet. I've only really got to the point, I've been trying to do the bottom up, trying to figure out what the mechanisms are suggesting, and then do some very simplistic top-down modeling. The bottom-up piece, if you start reading about pregnancy, the sort of dogma is it's immunosuppressive. But it turns out it's such a funky, complicated thing. It's much more than immunosuppressive. There's an awful lot of pieces going on. So I got that far, but I haven't got beyond that to also look at what that means in terms of whether males and females are super different in how those responses play out.
Great, thank you so much. I think we're going to end here. I don't see any more questions, but thank you so much for accepting our invitation and for your excellent talk.
Thank you.
For everyone watching, next week on Monday, our speaker will be Dr. Rob Salguero Gomez from the University of Oxford, and he'll be talking about life history theory and climate change, so don't miss it. Until then, have a great weekend, everyone, and goodbye.